All right, let's talk about this. We finally have some actual data on Omicron. These just got released yesterday, or I think it was 2 days ago. No, yesterday. So these just got released yesterday. There's 4 studies that were released. The first one is not really a study. It's basically a press release from Pfizer and BioNTech. And what they did is they don't have it's not a paper, so they don't have a material and method, so you don't know exactly what they did. But presumably they ran some neutralization assays, and we'll talk about how those work when we describe the other papers. And they found that a third. So what they found out is that there's a 25-fold reduction in neutralization titers against Omicron compared to the regular SARS, the before any of the variants, the wild type. And then if you give it a third dose, then it increases the titers by 25%, 25-fold. So you kind of bring it back to wild type protection with 2 vaccines. The thing that they also wanted to note was that most of the epitopes, which are the parts of the spike protein that are recognized by the T cells, are not affected in Omicron. So they think that T cell immunity should still be pretty good. So that's their thought process. But they're going to suggest a booster to protect you against Omicron because a booster seems to increase the titers to a protective level. And they make the vaccine, so they have some vested interest in promoting a booster shot. But the most interesting thing about this is I thought that the T cells are not affected by the mutations in the Omicron switches, so that's good. All right, so let's go through this other paper. This is out of South Africa, and of course they have access to the actual virus. So if you look at their materials and methods, what they did is that they got the virus from people that were infected by the virus and then grew them in these cells. And then they collected the viral samples from these SARS-CoV-2 Omicron variant cells the viruses they had collected. And so then this is how the live virus neutralization assay works. And this is how presumably all of them did it. So they took some cells that you can infect the virus with, and then they got plasma from people that had the vaccine. And then what they did is they would dilute the plasma. They would dilute the plasma and then they would mix it with the virus. So they would basically, they grew some virus, and then they figured out what concentration of virus gives you a certain number of infections in their cell culture. Then what they did is they took the plasma from people that had been vaccinated, and they diluted the plasma. So first they would put full strength plasma on there, and then they would mix it together for an hour at 37 degrees, and then they would put this mixture on some cells and see how many cells got infected. Okay, so that's pretty straightforward. If you don't put any plasma, then they knew that this amount of virus that they knew the amount of virus they were putting. They knew that it would cause, let's say 100 cells, 100 what they call plaques to form. And then if you put the full strength plasma, undiluted plasma, let's say that you only had 1, so you had 99% reduction. Then what they would do is they would just dilute this plasma. They would dilute it 1 to 10, 1 to 100. So they would be 1 part for 100 parts of just buffer. And then they would see how soon it would drop off. Okay? So that's how they're doing the thing. So basically, better plasma, you can dilute it more diluted, and it still blocks the virus from being able to infect the cells. Okay? So hopefully that makes sense to you guys. And so this is what they found out is here is they had 2 kinds of plasma. They had this orange plasma. The orange plasma is people that had gotten the BioNTech, so the Pfizer vaccine, okay? And then the green are people that got the Pfizer vaccine and they were also infected. So remember, vaccine plus infection gives you like super immunity, and it gives you really good immunity. And we had I will link the video where we talk about that up in a card like over there. Um, so what you notice is this is the amount of dilution. So like this is straight plasma. There's 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000. And then this tells you how far that you could dilute it and still reduce the amount of infections by 50%. So you see this BioNTech, this one person, they even at full strength, it did not block Omicron. And you see the Omicron ones, if you had BioNTech, if you diluted 1 to 100, that's when you lost 50%. If you had an infection plus the vaccine, you were doing better. And then, of course, everyone does better against the wild type. So the higher up you are on this graph, the more you could dilute the plasma and still block 50% of the virus. So higher is better. So you can see that this is better. The wild type is easier to block than the Omicron, and they said it's 41.4% harder to block this. And then also you can see that vaccine plus infection is better. It's higher than just vaccine. Okay, so that's the take-home message from that thing. This is a paper out of the Karolinska Institute in Stockholm, Sweden. This one's a little different. They did the same vaccine neutralization. So here's like 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000. And you see, again, the wild type is higher, so easier to block than the Omicron. And then you see here, the wild type is here, Delta is here, Omicron is here. So Delta is a little bit harder to block than the wild type, and then Omicron is even harder than Delta. Okay. The difference in this is that they used a synthetic virus in this paper. And you can tell, okay, let's see where they talk about that. I think, okay, here we go. Yeah, so Omicron spike pseudotype lentivirus. So what they did is they created a lentivirus, so not a coronavirus, but they put the Omicron spike protein onto it and used that as as kind of a test virus to check this. So this paper probably isn't quite as close to real life as the South Africa paper, which used the actual SARS coronavirus in their assay. So this last paper is from Frankfurt, Germany, and in this one, they used the actual Omicron coronavirus. So to evaluate the protective capacity, antibody-mediated neutralization efficiency against authentic SARS-CoV-2 Omicron was determined in vitro using an isolate obtained from a double Moderna vaccinated travel returning from Zimbabwe. So they found someone who'd flown back from Germany after returning from Zimbabwe. He had Omicron. They isolated the virus from him. They grew it up and they used it. And they did the same kind of neutralization assay that we talked about here. And then, so you see the same thing. Here is the gray is the wild type, and the orange is Omicron. So you see this is high. So this is 1 to 1, 1 to 10, 1 to 100, 1 to 1,000. So you see this is, again, the same type of dilution. And you see that this is 2 BioNTech, so 2 Pfizer vaccines. You see that's not very protective against Omicron. If you got a booster, then yes, protection increases. This is a booster 3 months out, so protection wanes a little bit after 3 months. This is someone who got infected and then got the vaccine. So again, you see that is also very protective, very similar to the first study out of South Africa, where you see that the booster plus infection is better. Okay. This is looking at Moderna, so similar picture here. 2 doses of Moderna, not very protective. Moderna plus a Pfizer booster is pretty protective. This is the AstraZeneca vaccine plus BioNTech one doesn't work very good. AstraZeneca plus 2 BioNTech or 2 Pfizer vaccines, that's pretty good. Okay, and the interesting thing that's different about this paper is they checked the monoclonal antibody therapy. This is the Regeneron monoclonal antibody therapy. And you see that, and then this is the dilution. So 1 to 1, 1 to 10, 1 to 100. So they're diluting the they're giving less and less of this Regeneron monoclonal antibody, and you see as you give less and less, the protection falls off. And this is the wild type and delta, the wild type and delta. But you see here, it has absolutely no effect on Omicron. So the Regeneron monoclonal antibody in the lab has no effect on Omicron. So we would expect in the patients that the Regeneron monoclonal antibody therapy will not work against Omicron. It does not seem to work. Okay, so now we have some good data on it. So far, what we know about Omicron is it does seem to be more transmissible. And we know that vaccine protection against it is significantly less, somewhere in the range of 20 to 40 times less. And then a third booster does seem to replace a lot of the protection against Omicron, according to these papers. And then we know that Regeneron monoclonal therapy does not work, at least in the lab, against Omicron. And then the other thing that we know is that if you have had an infection plus vaccination, then you're also better off. So infection plus vaccination seems to be as good or perhaps slightly better than a booster, a third, because like basically the infection is acting like a booster. Um, Okay, so I will link all these papers below in the description if you guys want to check them out yourself. But as expected, I mean, I'm always impressed at how fast these things are coming out. I know, this thing was just reported less than 2 weeks ago, and there's already papers. Of course, they're not peer reviewed yet, but people have done the studies looking at neutralization. We said that in 1 to 2 weeks we would have this kind of information, and indeed we do have it. So I'm very impressed at the speed that these labs are looking into these things. Okay, and then in a couple more weeks, we're going to get some real world studies about the efficacy of vaccines in the real world against actual infection and actual severe disease. Initial reports are that Omicron does not seem to cause severe disease, which is good news. Of course, it's too early to tell. There's not enough infections to really say that kind of information with too much certainty, but at least there aren't reports that make it seem like it's a lot worse. So my guess is it's similar or maybe a little milder than the previous variants, hopefully milder. I think that information will be forthcoming in the next month or so as the infections spread throughout the world and we get more information as more people are infected. All right, so hopefully this was interesting. I'll link the papers below. Any questions or comments, we welcome them, obviously. Thanks for watching.